So it was a pleasure to chair this meeting that took place yesterday afternoon and all day today. And I would like to thank two co-chairs who sat there with me throughout the meeting, some of them just for part and one of them for the other part. And this being a truly international conference, we had a chair from North America and a chair from Europe. And the two other chairs at this meeting were Bruno Balbi, who was chair this afternoon with me, and the other co-chair that I had was Giovanni Lungarella from Siena. So I think we had a good international representation, not only in terms of the participants of the meeting, the presenters and the discussants, but also in terms of moderating the meeting. Now I can tell you is that this particular international conference of alpha-antitrypsin was entitled Novel Concepts. Novel concepts in the mechanisms underlying the disease, novel concepts in how to detect the disease, and novel concepts how to treat the disease. And really what's most important to all of you who's suffering lung disease related to alpha-antitrypsin deficiency is, what does this mean? What does all this information that was exchanged the last day and a half actually mean to the patient? So it was easy to have the discussions about all of these aspects of lung disease in alpha-antitrypsin deficiency among scientists and clinicians. It's much more difficult to make sense out of all those data and relate that to you patients in terms of what does it mean to you, what we have discussed over the last day and a half. So I will try to do that, but I will have to go through all the individual presentations very briefly, and then I'll have some kind of personal conclusion that I would like to share with you. One of the first session was entitled Mechanisms, and the stage for this was set by Mark Brantley, who reminded us that while there are many variants in the alpha-antitrypsin gene, 95% of lung disease patients have a Z allele mutation. So that's still the most relevant mutant. He also talked about some other dysfunctional variants, so it's probably a little bit more complicated than just the S and the Z allele in terms of disease in the lungs. But it's still the Z gene that's really the most important in terms of lung disease. Now, Mark is from North America. So Sabina Jankiaskiin, who is from Germany at the present time, had some very interesting presentations, and this is something novel, very interesting presentations on the broad anti-inflammatory actions of alpha-antitrypsin as a protein. It's not only a protein that inhibits proteases, notably neutrophil elastase in the lung, which we know causes lung destruction, but it has a much broader anti-inflammatory action. It has an immunomodulatory action, and it has a much broader anti-inflammatory action. So it's a very potent anti-inflammatory protein in the body, which actually raises two issues. One of them is, how does alpha-1 antitrypsin augmentation therapy really have an effect on individuals who have alpha-antitrypsin deficiency in the lung? Does it do it by inhibiting neutrophil elastase or maybe other proteases, or does it do it by having this broad anti-inflammatory action? I think that's an interesting question. The other one is that if it does have broad anti-inflammatory actions, why couldn't it be used as a therapeutic agent in individuals who have certain diseases that are not associated with alpha-antitrypsin deficiency, such as cystic fibrosis, such as maybe COPD in individuals who do not have alpha-antitrypsin deficiency, which is the majority of patients who have COPD. So this really raises some very interesting questions going forward. The next presentation, again, still on mechanisms, was made by Stefan Marciniak from the UK. And he had a very stimulating and a very well-informing presentation on how the cell deals with the misfolded protein. As you know, alpha-antitrypsin deficiency is actually due to a protein misfolding problem. So the protein, alpha-antitrypsin, is not properly folded, and therefore it is toxic to cells. It induces all kinds of secondary mechanisms that can be toxic to cells. So he really dealt with the issue and all the intricacies of how the cells deal with the misfolded protein, especially in relation to alpha-antitrypsin deficiency. Then Jerry McIlvaney from Ireland, he pointed out how important the neutrophil, which is an inflammatory cell in the lung, is in terms of alpha-antitrypsin-related lung disease. And again, what he showed, and these are very interesting data, is that alpha-antitrypsin not only works in terms of inhibiting proteases, but it has many other effects on the neutrophil. And it actually reduces the action of the neutrophil in the lung, which in alpha-antitrypsin deficiency is a destructive action. Then we had Dr. Cosio, originally from Spain, then in Canada, and now in Italy, who pointed out, again, there is some other mechanisms of lung disease generation in alpha-antitrypsin deficiency other than protease, anti-protease imbalance. And that's autoimmunity. That maybe the polymers or maybe some other proteins in the lung that are present in alpha-antitrypsin deficiency work as an antigen for the body, so they're no longer recognized by the body as being their own, and the body then mounts a response to that, an immunologic response, which can lead to further lung disease. Then Irina Petracci, she addressed the question, and she is from North America, she addressed the question of whether or not animal models serve any function at all in alpha-1 research, particularly in alpha-1 related lung research. And I think her point was a very good one, because her conclusion was, at least my conclusion based on what she presented. My conclusion was that if animal models are used as bioassays to test an observation that are made in humans and to explain a human observation in humans with alpha-antitrypsin deficiency, then that animal model is very useful. But just to use the animal model as a model of disease is not the same as using the animal model for a bioassay to test something that you have observed in humans. So I think animal models will continue to serve an important function. Then we moved on to the second session, which was entitled Assessment. And Jamie Stoller from North America, he reminded us that it takes 6 to 7 years on average, and he's quoted several studies to show this, 6 to 7 years on average between first symptoms of lung disease and the final diagnosis of alpha-antitrypsin deficiency. He also pointed out that still in 2015, about 90% or maybe even over 90% of individuals who have alpha-antitrypsin deficiency, severe alpha-antitrypsin deficiency, remain undiagnosed. So the sobering part of his presentation was that if you compare these numbers 20 years ago to these numbers today, there has been no improvement at all. And then he suggested first of all, he listed some possible reasons for this. And actually had 3 reasons that he pointed out. One of them is the lack of knowledge of alpha-antitrypsin deficiency among providers. That's one. Another one is that maybe the guidelines should be made a little bit more user-friendly, so physicians, particularly physicians, will think of this disease a little bit more often. And then finally, that there is this nihilistic view that since there is no real treatment that works in alpha-1 antitrypsin deficiency-related lung disease, why diagnose it? So these are three of the reasons. And I think this nihilistic approach, again, feeds back into the lack of knowledge. So I think these three reasons are probably very realistic reasons. And then he suggested the remedies for this. And one of them is to provide free testing. And in the state of Florida, fortunately, we do have free testing. We don't have it in other states in the United States. And I don't know about other countries, but he thought that free testing may be an encouragement for the community to test more for alpha-1 antitrypsin deficiency. Another approach would be the electronic medical records, where you can put kind of a reminder on the medical records. If a patient meets criteria for COPD, why not put a reminder to the provider that maybe you should test for alpha-1 antitrypsin deficiency? And then finally is the involvement of allied health professionals in testing, in particular respiratory therapists in the United States and maybe in other countries, other professionals or allied health professionals who do pulmonary function testing, because that's really where the diagnosis of alpha-1 antitrypsin deficiency lung disease is made. Then, as far as assessment is concerned, we moved on to David Lomas's presentation. He will talk to us later today about something different. But he showed results from a to me, very interesting study that GlaxoSmithKline, a pharmaceutical company, a giant pharmaceutical company, decided to do an observational study in COPD, not an interventional study. They did not test one of their products in patients with COPD, but they just wanted to know what is the natural history of COPD. So they sponsored this very expensive study, which has generated a host of new findings that we did not know before this study was done. And what David pointed out, that the study actually started identifying some biomarkers for COPD. He also pointed out the importance of exacerbations in the progression of lung disease in patients with COPD. And interestingly enough, they also started looking at some of the genes that may be associated with COPD in general. Now, I'm not talking about alpha-1 antitrypsin-related COPD. And one of those genes that came out is a gene that may mediate nicotine addiction. So that was an interesting twist in his presentation. Then we moved on to Rob Stockley from the UK. And the gist of his presentation was to show that FEV1, you're probably all familiar with FEV1, which is this test where you take a deep breath in, you blow out as much as you can, and then you measure how much volume comes out of your lung, from the top of your lung down to the bottom of the lung, in 1 second, in the first second of this forced expiration. And FEV1 is a measure of lung function that is done worldwide. It's relatively easy to do it, and almost all laboratories will do that. And it has become a standard for assessing pulmonary function, including in COPD. But he pointed out that there may be a better test to look at lung function, and that is actually looking at the uptake of gases or the exchange of gases in the alveoli. So this is the lung tissue where actually gas exchange occurs. FEV1 doesn't measure that. FEV1 measures the ventilation, more or less, of the lungs. But the gas exchange itself is a different function of the lung. And there is a test, it's called well, we call it diffusing capacity. In England, they call it gas transfer. It's essentially the same test. But he pointed out, in his studies, he found that this gas transfer actually might be a more accurate way of looking at lung dysfunction in patients with alpha-1 antitrypsin deficiency. And he also mentioned something else. As you know, lung function measurements, lung structure measurements by themselves don't really fully describe the disease and don't fully describe what a patient with obstructive lung disease experiences. There is this health status or disease-related quality of life that also has a major influence on patients. And what he showed that if you look at disease-related quality of life and you assess that with metrics that exist for it, that those patients who interact with their physicians well have an improved quality of life just on the basis of interacting with their providers, which I think was a very interesting observation. Then we had Jerry Turino from North America, and he talked about biomarkers, and he particularly talked about the biomarker that he developed for monitoring lung disease in patients with alpha-netotrypsin deficiency or patients with COPD in general. And I would call his biomarker an intelligent biomarker, because it's a biomarker that actually relates conceptually to the destruction of lung tissue. So this is a biomarker that actually looks at breakdown products from the lung as the lung is getting destroyed. You can measure it in blood, you can measure it in urine, you can measure it in sputum, you can measure it in bronchoalveolar lavage. And he found that in alpha-netotrypsin deficiency, individuals who get augmentation therapy have a lower level of this biomarker in their blood and in other samples that he measured than individuals who do not get alpha-1 antitrypsin augmentation therapy. So that was a very interesting presentation. Now, I would like to point out that other biomarkers have been looked at in COPD, but some of those other biomarkers are not conceptually easy to understand. They correlate with lung disease, but in terms of mechanisms, it's sometimes difficult to figure out why. In this particular case, that's why I call it an intelligent kind of biomarker. We know exactly why this biomarker relates to lung disease. Then we had Dr. Stoel from the Netherlands. He talked about lung density. As you know, CT scanning of the chest has been used as an outcome in COPD in addition to lung function. And he talked about many technical aspects of that and kind of told us that the field has advanced a lot, so I think we should be more and more confident in actually using CT scanning of the chest as an outcome in COPD, including COPD in patients with alpha-netotrypsin deficiency. It's essentially looking at the structure of the lung. FEV1 and gas transfer looks at the function of the lung. CT scanning looks at the structure of the lung. And we know that the lung gets destroyed in COPD, so why not look at the structure? So that's, I think, is a very important outcome. Then after him, we had Ed Silverman, who is from the United States. And he talked to us about the risk of lung disease in heterozygotes. Now, this is something fairly novel, and he showed data which are pretty convincing, to me at least, that individuals who are heterozygotes, and don't forget there are many more heterozygotes than severely affected alphas, but heterozygotes have an increased risk of developing lung disease, particularly if they're smokers. So this is a fairly new observation. This has been looked at for many, many years, but I think finally we have some convincing data that indeed that is the case. So that was quite interesting to hear. He also talked about many other genes that have been associated as part of the COPD gene study that he is actively involved in. But I think this finding of the MZs actually having an increased risk of lung disease is a very important one. And then we moved on to treatment. And that was the last session. The first presentation was made by Dr. Grisi from Germany. He talked about the use of inhaled alpha-antitrypsin for the treatment of lung disease. And I think the conclusion from his presentation is that, number one, inhaled alpha-antitrypsin is safe, as far as we know today, from the various studies that have been done. The second is that alpha-antitrypsin by inhalation has been used, and the data have been reported for CF, cystic fibrosis. And it has been shown that on biochemical outcomes, not clinical outcomes, but on biochemical outcomes, inhaled alpha-antitrypsin does what it is supposed to do in patients with cystic fibrosis. Studies are ongoing in patients with alpha-antitrypsin deficiency, and those results are not known yet at the present time, but hopefully they will be positive as well. Then Dr. Jan Stolk from the Netherlands, he talked about cell-based therapies. He indicated that we are not there yet, but cell-based therapies, stem cells, are being used now to replace a damaged lung tissue. It's going to take some time for those to become clinically useful, but a lot of progress is being made, so I think that was very encouraging. But he also pointed out that in terms of cell therapy, the liver as a target is a more likely target to be successful in the near future. The lung as a target is something that is going to be lagging behind the liver. And then Ron Crystal from the United States, he talked about gene therapy, and he discussed the various techniques that have been used for gene therapy. One of them, and Mark is going to talk about that, is to transfect skeletal muscle to produce alpha-antitrypsin and raise its level in circulating blood. Another one is to use the liver. And another one that Dr. Crystal is using is actually the lining of the lung. So what he's doing is he's putting these vectors, these viral vectors, into the space between the lung and the chest wall, which is called the pleural space. And he transfects the cells in there. And the advantage of this technique, although we don't know the results yet in humans, but the advantage of this technique is that you're actually transfecting tissue right near the lung or in the lung, as opposed to transfecting tissues outside the lung. So we'll see how that comes about. He has already shown that this is safe in animals, and he has shown that he's able to raise serum alpha-antitrypsin levels in these animals significantly by using this lung lining gene therapy. And then the next presentation was made by Sandy Sandhouse. He talked about the various studies that have been done to look at the effects of augmentation therapy on lung disease in patients with alpha-antitrypsin deficiency. He pointed out that the endpoints that have been looked at were lung function, lung structure. We already talked about those two endpoints. And finally, mortality. And if you look at all of these endpoints, collectively, if you look at all the studies that have been done, I think the conclusion can be drawn, at least by me safely today, that augmentation therapy does have a beneficial effect. And then the final presentation was made by Dr. Mark Forshak, who is from industry, and he talked about the trials and tribulations of registering alpha-antitrypsin products with regulators. So if we academicians think that doing research is complicated, what we heard from industry, it's manifold more complicated than what they have to deal with. So that was, in a way, sobering, but at the same time, he was very positive in his presentation. And the most positive part of his presentation, to me at least, was that he said that even big pharma is becoming more interested in rare disease research and developing new treatments for rare diseases. And let's face it, alpha-antitrypsin deficiency is one of those. So I think that was a very positive remark that he made. So in summary, what did I take away from this? What was my impression in terms of what novel things have occurred in terms of alpha-1 related lung disease? So I came up with 6, and I'm sure there are others, but I only came up with 6. One of the novel things that really struck me was the increased risk of lung disease in carriers. That's fairly novel. At least we have more convincing evidence now for that than we had in the past. That augmentation therapy slows the progression of lung disease in alphas who have lung disease. That alpha-1 antitrypsin has this broad anti-inflammatory effect, which opens up new avenues for its use beyond augmentation therapy in patients with alpha-1 antitrypsin deficiency. The next point is that inhalation of alpha-1 antitrypsin in terms of a therapeutic intervention is safe, and maybe in the near future we will hear some more information on whether or not it's truly effective in slowing the progression of lung disease in patients with alpha-1 antitrypsin deficiency. But it certainly is an attractive approach to treating such patients. I also think that particularly the mechanistic approaches that are being used to understand alpha-1 antitrypsin deficiency have made a lot of progress. And they will serve, I think, going forward for new drug development, because I think new drugs and new principles of treatment will have to be based on a better understanding of the mechanisms. And we have learned some new things about mechanisms that will help that process. And then finally, I think we are closer to gene and cell therapy, closer in the liver than in the lung, but I think we are getting closer to them being eventually clinically useful. So overall, I think we should have a positive outlook for new treatments. At least that is my feeling after having participated in this meeting for a day and a half. But I have the feeling that if we don't get it tomorrow, we will get it the day after. So thank you very much.